Well, thanks everyone. Good evening. I'm really overwhelmed actually and very touched that you've all came here on a pretty cold December evening and to support our research. And I'm really looking forward. Hopefully you'll enjoy what I'm going to tell you. I'm going to tell you a little bit about my journey into cancer research and some of our main findings. So I thought I'd start off with stress. I think every academic in the room and definitely the PhD students know what stress is. But actually, we all discuss stress and we often say, you know, you may hear the term, oh, I was really stressed out and he ended up getting the flu or he ended up getting a bad cold. And that's right. We know that stress, you know, affects our immune system and it can make us more susceptible to colds and flu. But as a cancer biologist, I'm hoping to try and convince you today about the link between stress and cancer and what we've been working on for the last few years. So stress is something, just to get us all on the same page here, stress is something that we experience when we can't cope effectively with a situation. So we may all have stressors, but how we perceive that stress and how we cope with it is quite important. And sometimes you may see a situation as mildly challenging. So you're late for a meeting or late for a bus, and that's good for you. That's your fight or flight response. You'll have stress hormones released and you know you'll be fine. But the stress that I study and the stress that I'm interested in is that stress, that overwhelming stress when you feel you can't cope with a really tough or threatening situation, which could be, for example, a cancer diagnosis. And so I've got a picture here of this lion here. Obviously, this guy here is quite stressed at the moment with a big lion chasing it. But it really is that chronic stress. And I do want to distinguish between the two because short-term stress or worrying about an exam is good for you. But actually, it's the long-term, those significant stresses that we're really interested in as cancer biologists. So what happens when we get stress? So basically our brain registers that something that we find is stressful. And what happens is our adrenal glands will produce stress hormones. So as James said very nicely, the stress hormones that I study, they are called glucocorticoids or cortisol. You may have heard the term cortisol, and also catecholamines like noradrenaline or adrenaline. You might hear if you're running for a bus or something, your adrenaline levels increase. And so I'm really interested in these stress hormones. And what happens is when these stress hormones are released, they bind to receptors on our cells. And almost all of our cells in our body have these receptors. So it means that every cell in our body pretty much can respond to a stress hormone. This can be a normal cell, a cancer cell, an immune cell, for example. And then when the stress hormones communicate or bind to a cell, then they'll alter our genes and proteins and they can tell those cells to do different things, to move and migrate. So that kind of is what happens when we get stressed, and that's what I really study, the stress hormones. But I want to take you back a little bit and start where it all began. So this is me in the 70s, the era of brown flary chords, flower power. I'm not going to say when in the 70s, loosely we'll say 70s. And this is my lovely mum and dad, and who are absolutely amazing. They're here tonight and they're here to support my research. They've been absolutely brilliant. And so, and as a child, apparently, I asked a lot of questions. As where people would say, stop asking questions. How does this work? Why does this do this? How does it do this? And I didn't know at the time. I guess it's a scientist in the making, really. And so it wasn't until I went to Chellaston Senior Schools. This was me at 15, I think, 14, 15. And I really got interested in science. And this will kind of show you how old I am. At the time, we weren't allowed to study the three sciences, chemistry, physics, and biology. But that's what I loved. I loved the sciences and I love languages as well. And this was us on a French trip going to France. And so I had a couple of issues because I knew I needed the sciences to do what I wanted to do, but they wouldn't allow us to do three sciences. And I also wanted to be in the top set of physics. I got the grades to be in the top set, but the physics teacher said at the time, no, I'm sorry, we can't have girls in the top set. I said, well, why not? I know you wouldn't hear of that now, would you? Why not? Well, anyway, I came home to my mum and dad and I said, you know, I want to do the three sciences and I think I should be in top set for physics, but, you know, it's a bit challenging. So my mum, 5'1" of her, went into that school. I don't know what was said, but when she came out, I was doing the three sciences and got in the top set of physics. So And then I managed to do the 3 sciences at A-level, and then I did, as James said, I did an undergraduate degree in biology at the University of Portsmouth. And actually this was quite interesting. A lot of people focused on marine biology, but there was about 15, 20 of us that liked the genetics and cell biology side, and I absolutely loved it. And I was thinking, yes, I think I love it, and I think this will be great for my career. I just really love cell biology. And at the time I wanted to be a vet, and I actually did get a place at Edinburgh University to study veterinary medicine, but the costs were just so high and it was such a long time. And so I was in a little bit of a quandary, and a wonderful professor took me aside and he said, I am running an MSc in applied toxicology. It's funded by Glaxo Wellcome. There's 1 position and there's 10 people applying. I think you should go for it. So bless him, I went for it and I interviewed and I got the position, and absolutely love research. The best thing I ever did, because I got to go to work at Glaxo. I worked at Pharma for 3 months, but I could also learn about drug behaviour and toxicology as well. And that was really amazing. So people that know me know I love mentoring, and if you're in my lab, you will be mentored. And as Renee, she is my first PhD student in the UK, she's sitting here in the audience, she said, Mel will mentor you whether you want it or not. And I'm all right with that. I'm actually okay with that. So what I thought I'd do is take a bit of a gamble, and I'm going to do a few mentoring tips during my inaugural. I just got to do that. And my first mentoring tip is obviously find yourself a good mentor. So mentors are absolutely amazing, and I've had lots and lots of mentors in my career, and I'm just highlighting a few here. And I'll talk about these as we go through. But Sally, when I was at the Centre for Disease Control, she was great. She taught me how to write and focus and build my independent research. Steffi Österreich and Adrian Lee, absolutely phenomenal mentors. I saw what they did in Pittsburgh, how they helped me with my career to bring mechanism to my career. Nancy Davidson wasn't a direct mentor of mine. She was the head of the Cancer Centre, but she's given me a few tips that I'm going to share with you along the way. And everybody knows here Professor Dame Leslie Fallowfield, who is trying still, I think, to mentor me on how to communicate better, and that's what I'm trying to do. So mentoring is really important to me. So after I did my MSc, I did a PhD in biochemical toxicology at Imperial College, and this was also funded by AstraZeneca. And it was here that I really started to get my love of the immune system. So we were studying at the time allergic contact dermatitis, and I was interested in one, it's a cytokine, it's a signaling molecule that activates and talks to cells. And what we found was that this signaling molecule played a really important role in skin responses, and it help dampen down some inflammatory responses. And so I really got into the nitty-gritty of the biology, but I got into immunotoxicology, so really learning about our immune cells, how our bodies can fight disease, and I really, really enjoyed that. And I got my first paper published here in this journal, Cytokine. It's a small journal, but I was ever so proud and chuffed of this paper. And I think the PhD students, you always remember your first paper when you get that acceptance letter. And I was really thrilled. And then this was, we all went to the British Toxicology Society, and we all went out there to celebrate. So that's how I worked on my PhD. And it was interesting, after my PhD, I had a couple of choices to decide what to do. I was looking for postdocs, and I had 2 opportunities. And one was to go to Charing Cross and study HIV. And I believe Elton John had just donated a significant amount of money to the lab. And I thought, cool, it's the immune system. But the other one was to go to America to work for the CDC, still in allergic contact dermatitis, but to start studying stress. So I thought, I love to travel. So I chose America. And so this is the exact photo actually I sent my mum. I said, here's the window of my office here. And, you know, because I think I did consider Australia at the time, and my mum said that's too far. So she was all right with America after that. This was in West Virginia, and this was under Sally Tinkle. And what I did here was, the idea was, it was for the National Institute of Occupational Health and Safety. And the goal was that they thought that stress and stress hormones can exacerbate skin diseases. So if anybody here has eczema or you have psoriasis, you may know when you get really stressed that you may get flares and triggers. And my job was to come in and look at the mechanisms and see really what stress did. And so I did a large body of work, but I don't want to focus on that because I want to get to the cancer work. But what we found was that stress could actually change our skin cells. So these are Langerhans cells that sit in the skin, and if you have an allergen on your skin or something like that, and you see a red blob on your skin, what will happen is cells will go to your lymph node, talk to the immune system, and then you'll get an inflammatory response, an immune response. And we found that stress changed the shape of our cells and also altered the ways that cells could migrate and move. And at the back of my mind, I was always thinking, I always wanted to go into cancer research, and I'll tell you the reason why when I get to my cancer slide, but I was always thinking about cancer research. And I was thinking, well, if stress does this to our skin cells, then what could it do to cancer cells? So I started to really start forming these ideas. And I also got a Mazda Miata when I was in America. Absolutely love this car, and it was a convertible, and in America they have these beautiful big roads that you can drive up and down, so I did have a lot of fun with this. Anyway, I did work hard. I spent 5 years at the CDC in between driving the car, and And then I was really thinking about cancer more and more, and I thought, a part of me was kind of getting fed up with people showing me different bits and bobs on the skin. I thought, I actually want to go into cancer research. And so what I did was I contacted a guy. This is a picture of beautiful city of Pittsburgh. And here you can see it's built on 3 rivers, and there's UPMC, the University of Pittsburgh Medical College. So I contacted a guy called William Chambers, and I said, I study stress and allergic contact dermatitis, but I really want to get into cancer research. I lost someone very dear to me through cancer research, and I want to study it, but I want to study stress and cancer research. So I sent this email to him. He emailed me back a day later, and he goes, "Hello, Melanie." He said, interestingly, I was having a cup of coffee with Andrew Baum, a biobehavioural scientist, yesterday, and we were just saying that we should build up a programme of stress and cancer and the immune system. And so, cut a long story short, I interviewed over there, and I got the position at the University of Pittsburgh Cancer Institute. My mentoring tip number 2. Be brave, okay? So if you're a PhD student, you're going out for your postdocs, if you like a good lab and you see somewhere you want to work, cold call, send an email, you never know what might happen. Or offer to write a fellowship, perhaps, something in their field. So this is something, don't be afraid to contact future employers. And I was very lucky I had that opportunity. So at the time, the Cancer Centre was led by the late Ronald Herbermann. He's a fantastic guy. He was one of the guys that was involved in the role of natural killer cells in fighting cancer. And here was the building that I worked at, the Hillman Cancer Centre. And it was absolutely brilliant because when you walked in the entrance of the Cancer Centre, he designed it very well, that we walked in the entrance together with the patients. The scientists went to the right, the patients went to the left, but every day you walked in with patients. And the late Ronald Herbermann knew what he was talking about. He says, I want you to see patients every day. I want it real for what you do. So we all worked long days there, as you can well imagine, and long weekends. And so it was there that I really wanted to look at how these stress hormones, these glucocorticoids and catecholamines, could influence diseases such as cancer. And here I've got a diagram. This is from a publication, a more recent publication actually. But what happens is when we have cancer cells, these are cells that keep growing and growing, and we don't want that. These cells are able to hide from the immune system. They cause inflammation and tissue damage. They can move from the primary site to secondary site. So in the case of breast cancer, which is one of the cancers I study, if you catch cancer early, that's great, but if not, it can move to the bones and the brains and the lungs, and these are really hard to treat places. And so this is what I set up at the Hillman Cancer Centre. And I started off, I thought, well, I've worked on the immune system, so let's start with the immune system and see what stress does to our immune system. And what we did was we took blood samples from happy, healthy people, and also from people that were very stressed. I think it was volunteers of students undertaking exams, and they were stressed. And what we found was that actually stress does damage our immune cells. And we've known for a while that if you take a blood sample from a stressed person, you'll see less numbers of immune cells. But what we did at this time was identify how stress can affect the immune cells. Because if we want to get clinicians on board, and we want to really encourage clinicians of how bad stress can be, not this kind of anecdotal, we're all feeling stressed, but it has these physiological effects on our body, then we need to highlight the mechanisms. And so we highlighted a pathway here, which we won't need to go into today, but through how stress can damage our immune cells. But all the while, that was fine. We know the immune system plays a role in cancer, but I was also really interested in how stress hormones induce DNA damage and impact repair in cells. And what we did was we got some 3T3 cells. 3T3 cells are not cancerous cells, but they are cells that readily transform. So I thought, if I don't see anything with these cells, we're not going to see anything with normal cells. And the question was can stress hormones cause DNA damage in these cells? So what we did was, this is DNA damage. So the higher bar here means there's DNA damage. This is a control, so UV light, for example, and we see that UV causes damage and it can be repaired. So the good thing about DNA damage, it can get repaired, unless you have deficient repair pathways, but it can get repaired. And what we found was if we added stress hormones, cortisol or catecholamines here, that we saw an increase in DNA damage. But the good news is if we have these receptor blockers, because remember I said that our cells have these receptors that make them receptive to the stress hormones. If we block those, then we can reduce the damage. And we also did another assay, a cellular transformation assay. So cells like to sit together. If we grow them in a dish, these are just cells that we grow in a dish. We can just buy them online and grow these in a dish. And if we grow the cells, they don't like growing sitting apart. So if you grow them in soft agar, they won't grow. If they're normal cells, they won't grow. But if they start to become cancerous or tumorigenic, then they will start to grow. And we found that those cells treated with stress hormones started to grow just a little bit. And again, we could reduce this with the stress hormone blockers. So this was the first idea that stress can really have an impact, perhaps, on these cells. Again, these are precancerous cells. So going back a little bit to cancer incidence, so in the UK, 1 in 2 of us will maybe diagnosed with cancer in our lifetime. So it's something that we can, expect, but hopefully it's something we can treat and then it becomes a chronic disease and we can live with this. And in breast cancer, for example, 1 in 7 women may develop this in their lifetime. And here is a picture of my beautiful Auntie Jean. So she is the reason that I do cancer research. So my Auntie Jean, she's not my real auntie, but she was a neighbour that kind of lived next door and raised us. And she passed away from metastatic breast cancer when I was at university, and it had a massive effect on me. And I thought, okay, we need to figure this out. How did it happen? What can happen? She wasn't super old, and this is her husband, my late Uncle George as well. And so that's the reason. So any PhD students in the audience or any students is that you have, it's what drives you, it's passion. Cancer research is not a 9 till 5 job. It's something that you've got to love and really enjoy to do. And that's the reason that I really got into cancer research. So we've spoken a little bit about stress, and I've told you how I got interested in the immune system and stress in the immune system, but now we kind of want to bring it together with cancer. And the National Cancer Institute report that about 60% of patients feel anxious when they have go through their cancer trajectory. I think that's a low number, actually. I think it's everybody. Leslie's nodding at me, and I think she knows her stuff, so we're going to go with that. And then about 30% is significant anxiety. 20% can lead on to depression. And then I think it's 10 to 15% have reported post-traumatic stress disorder, which I found really fascinating, because as far as I'm concerned, I heard of that when you hear of people going off in the army and they're fighting the wars and seeing terrible things. And I'm thinking, blimey, if cancer patients are reporting, even 1 or 2 cancer patients are reporting this, then that's quite a stressor. So not one just to sit and read statistics. I thought, well, I'm going to actually chat to cancer patients and find out what they think. And so what I did when I was in America was I asked cancer patients, what are the key, I really wanted to know, do they feel stress, and can they share with me their key stressors? And what they said to me, their key stressors are obviously a cancer diagnosis, being told you've got cancer, which is quite scary, and chemotherapy, particularly if they were young women with breast cancer. Will I lose my hair? Will my who will look after my children, and so on. And finally, and the one that I thought was really interesting, was the fear of recurrence at the end. So at the end, when patients are told you're in remission, I thought, that's let your hand off the wheel. Things are great. But actually, that's still a stressor, and there is that worry there that it may come back. And it varies between different people. But I thought that was very interesting to me. So I thought, okay, this is something that is serious, and my job as a molecular biologist is to try and figure out, if these patients are reporting stress, then what is stress really doing to their bodies? And so obviously, so when people are so cancer patients or people living with cancer, they're not just people living with cancer, they're also undergoing drug treatments. And at the time when I was at the Cancer Institute, there wasn't really many immune therapies at the time. It was mainly chemotherapies. There were some immune therapies, but these very toxic cytokine therapies that we're looking at in melanoma, for example. And with chemotherapy, I thought, we know that patients respond to chemotherapy, but some patients can become resistant to them. And what that means is you have cells, you can add a chemotherapy drug and you see the cells die, but then you can add the drug again a month or so later and the cells don't die because they're resistant. And as a toxicologist, I thought, well, this is really interesting. I thought, well, you know, if patients' cancer don't respond to treatment, and we know that stress hormones can affect our cancer cells, we know it can affect our immune cells, can it affect drug treatments? So I phoned up Sally Tinkle, who I work with at the CDC, and I said, I've got this idea that stress could impact drug resistance. What do you think? She says, yeah, I like it. I said, you don't think it's too off the wall? And I did a literature search, and there was only, as far as I could tell, only one other lab at the time that was looking at this. Was a lab in Chicago, Susan Conson, who's fantastic and who we've collaborated with. So I thought it's not too off the wall idea, but it's something that I can really study and really hopefully help patients with as well. And so my mentoring tip number 3 is try and become independent early with your thinking. So if you're a PhD student in the audience, I know a lot of you have ideas. So my job as a supervisor to PhD students is to help you get your PhD and train you and get you excited, but also to encourage you. If you have a little idea, we call it a gardening project in our lab. You want to do a little project and something to build your ideas up. And also get feedback on your ideas. And people can be brutal. Have a brutal, critical friend. They're my favourites, okay? You just might need a glass of wine when you visit, but usually they help you. And it's really, really important to do that. So then what I wanted to do was to move, to gain my independence a little bit. And at the time, there was a chappie called Tom Conrads, who was a mass spectrometrist, and he moved over to Pittsburgh and he wanted to set up a proteomics lab. And we got chatting. He said, you're a cell biologist, molecular biologist? I said, yes. He goes, so what he did was he offered me a great deal. He said, if you come and be a cell biologist for my proteomics lab and help run the cell work and do some mass spectrometry, you can have 50% of your time to develop whatever program you want. And I thought, fantastic, this is job security. I was in America at the time. And so I started to do some really nice research using mass spectrometry. And here what we did was we got some cells and treated them with chemotherapy, just paclitaxel. We got cells with chemotherapy and we added just stress hormones to the mix. And then we did a technique called SILAC mass spectrometry. But what we found was that the stress hormones were triggering drug resistance genes and they were kind of triggering proliferation genes. So they were telling our cells to proliferate and grow fast. So I thought, okay, this is something we can really start looking at. And we wanted to look phenotypically. So what do stress hormones do to drug treatments and cancer cells? So I'll talk you through a little bit of data here. So here is, this is just a proliferation assay. So the higher the bar here, the higher the number, the more proliferation the cancer cells. These are 2 different types of cancer cells here. They're both breast cancer, but different types of breast cancer. And what I did was I worked out the concentration of paclitaxel to kill half of the cells. So that's great. That's what we want with chemotherapy. We want to kill our cancer cells, right? And then I thought, well, I'll add in stress hormones and see what happens. And if we added in cortisol or our catecholamines, you can see that the cells were proliferating again. And so what that means is that the stress hormones are interfering or somehow causing drug resistance. And we did a lot of work on this, and we ended up showing it through a DNA damage pathway, and it was telling the cells to halt in the cell cycle. And we know that chemotherapy acts on rapidly dividing cells. That was one of the mechanisms that we're looking at. But the good news is that there are receptor inhibitors here that can block some of these responses, because I think I did say in my advert here that we'd talk about the good things that we can do about stress. It was around this time that I got married, actually. And you might recognise Dr. Nick Stewart there. My mum made him wear that top hat. He didn't volunteer that. And actually, and I mentioned Nancy Davidson, and actually at the time, Tom Conrads was leaving to go to a different place, and I was very worried about my position. I ended up staying at Pittsburgh in the pharmacology lab, which was fantastic. But she said to me, she says, Melanie, this is the most important time of your life. Don't worry about anything else or anybody leaving. Just focus on your day of getting married. And he's still with me today, so I think we've done something right. And then Steffi Osterreich and Adrian Lee came to the Pittsburgh Cancer Centre, and I'd seen them a few months before because they'd given a talk, and I was absolutely really impressed. A phenomenal, dynamic duo in breast cancer, and what they don't know about breast cancer, quite frankly, isn't worth knowing. They are amazing, amazing mentors. And so I started to work here. This is McGee Women's Research Institute, and we started to really look at how stress can influence cancer and diseases. And I was looking, this is one example, so I was looking at immune trafficking. So when you think of cancer cells, there's also the immune microenvironment together. It's not just cancer cells growing, but there are structural cells and immune cells in there as well. And so we really started to look and see what stress hormones were doing. And it was here I got my first grant, actually. I'd already, when I worked for Tom Conrad's, I'd put in a Department of Defence grant, and I got a score of 1.1. So the highest is 1 and the lowest is 0. And I was really chuffed. I didn't get the money, but it was really, really close. And it gave me confidence that even though I didn't get that grant, that I knew I could get funding. So anyway, this was just a small grant. It was just $50,000. But I was so chuffed when I got this grant. But what happened was, it was in the days where we had to print off the grants and we posted them off. And so Paul Nadine at the time, who was the administrator for Steffi and Adrian, was posting all of these, printing off these copies of the grant. And I saw another copies on the side. I said, oh, who's this at? She said, oh, it's Steffi Osterreich. She's putting a grant in for the same place. I said, oh, don't bother putting my grant in. She says, why not? I said, because funders don't often give 2 grants, particularly small funders are not going to give 2 grants at the same institution. Anyway, she says, don't worry about it. I've printed them off. Let's get them sent off. And as you can see from here, both Steffi and I got the grant, and we were both really chuffed about that. And this was my first grant on stress and drug resistance. And based on feedback from the DOD grant I wrote, everybody said this is an important area of research. It's an understudied area of research. So I knew I was on the right track here to study this. And so my mentoring tip number 4, I said I'd keep doing this, and I apply for all the funding that you can. And even if it's just a little grant of $50,000, that can turn into something else, and it's really worth doing that. And diversify your funders as well. You know, try charities, and there's UKRI and companies as well. It was around this time we had our own little stressor. So, sorry, Ollie. So Ollie joined the gang, and I also want to share with you some advice that I got also from Nancy Davidson. So I only had 3 months maternity leave. I had Ollie, I went back to work, and about 3 months later I did a poster on my stress and drug resistance. And Nancy Davidson, who was head of the cancer centre at the time, stood quietly there. So I was talking to a very enthusiastic Russian immunologist that was so keen and asking me lots of questions on my poster, and Nancy just stood there quietly waiting. And so I thought, oh my gosh, she spotted a fundamental flaw in my poster, and that's why she's coming over. Anyway, it wasn't that. She just came over and she says, I want to congratulate you on your birth of Oliver. That's really great news, and the best thing in your life-changing thing. And I thought, what great advice. That was absolutely really nice of her to do that. And so we, and then under Steffi and Adrian's mentorship, we thought about really looking at breast cancer. And Steffi says, okay, so you say that these are receptors on every cell. Let's look at, Nancy Davidson's got a panel of 39 breast cancer cell lines. Let's see if they really do have these receptors and if they're doing anything. And so what we did was I screened just by PCR. These are different types. These are cell lines. So these are cancer cells that were originally taken from patients, and we can grow them and culture them. And we just screen them for stress hormone receptors, the glucocorticoid receptor and the adrenergic receptors. And what we found was that almost all the cells express the receptor, but importantly, one type of breast cancer, so I don't know if you know this, but breast cancer can be divided into different clinical subtypes. And one of the types is triple negative breast cancer, so it doesn't have estrogen receptor, progesterone receptor, or any of these receptors on the surface. It's normally more aggressive. It can be associated with patients with mutations in their BRCA genes. And what we found was that the higher levels of stress hormones were in this triple negative cell line. And then these graphs here are just showing that not only do those cells express those receptors, but they also are able to signal. And so we really started to look into this. I thought, okay, these stress hormones really are there. They seem higher in aggressive cancers, and we now know some of the more aggressive cancers in different types of cancers, like endometrial, for example, that we think that they have higher levels of these stress hormone receptors. And my mentoring tip number 5 from Nancy's advice is manage your time well. It's really important to have a good work-life balance. So obviously I had a young child at the time and I was working full-time, and it's really important to have friends. And these are my friends. I've got a couple in the audience, Debbie and Jenny here. These are what I call my mum friends, so friends who have children in the same year. They are a lifeline. It's ugly sweater day on Monday, odd sock day. We've got to take crackers and cheese on Monday. It just really helps us having this group, and I'm massively indebted to these guys to help me out. And then this is my best friend Jo, who's been a massive supporter of our cancer research. I've known her since I was 7, and it's really good to have that work-life balance as well. So I spent some time in Pittsburgh, and I really learned a lot under Adrian and Steffi, and we published quite a few papers looking at how stress can impact cancer and cause drug resistance. And about that time, Ollie was about 2 at the time, and I remember saying to Nick, I'd like Ollie to be with family. So Nick's Canadian, so I said either we go to Canada or we can come to England and do some work and be with family. And ultimately we decided on England, and we interviewed here. We did at different places. I interviewed at the Van Geest Cancer Centre in Nottingham and here, and it was so good here. And Brighton was very attractive for me for a couple of reasons. 1, it was a very collegial environment. 2, there's a brilliant med school up the road. Leslie's group I knew was phenomenal, and I know I can't do my research without Leslie's group. I need psychologists on board. I am not a psychologist, and she has taught me so much, actually. And so we decided to come here. And actually, this is a slide from my interview at Brighton. And this was my vision for the lab, was I said, so I do a lot of lab work, and I may talk to patients, but most of my work is usually on cell lines and looking at stress hormones and looking at signaling pathways in our immune system. But what I really want to do is help patients. I wanted to see that patient benefit. I want to translate my research to help patients, and that was my goal moving here to Brighton. And so and I had ideas at the time to work with Shaw-C and to look at patients, and so I could share with you some of that work now. And obviously you'll all recognise this as the University of Brighton here. And so when I first started, and I'd only been here a couple of days, there is a lady here with glasses, and she is in the audience, and she won't mind me saying this, everywhere I went she was there, Renee. I'm really interested in breast cancer. I really want to study breast cancer. And she was so passionate about breast cancer. So what we did was, together with Bavik, who I think is here in the audience somewhere up there, who's a electrochemist. So I had an idea of a project, was that we knew that stress hormones could cause DNA damage, but we didn't know how. And I thought it might be through reactive oxidative species. And so together we cooked up a project for Renee to have a look and see if stress hormones such as cortisol can increase reactive nitrogen species. And Renee did a lot of work on this, and she found that cortisol increased the production of nitrite, which is a byproduct of nitric oxide, and which can lead to DNA damage here. And also she looked at clinical samples and invasive breast cancer carcinoma, and found that INOS was upregulated in these breast cancer samples, and she looked at cancer cell lines as well. So she was really looking at this different pathway of how stress hormones could potentially cause DNA damage. And finally, knowing my love for the immune system, she she developed this model where she co-cultured cancer cells together with different types of immune cells, such as macrophages, where she could study the effects of cortisol and figure out, really how bad cortisol can be and what cortisol can do. And it can kind of, using these macrophages, cause this kind of pro-tumorigenic profile, which isn't great. But again, we can do something about stress, right? And so a long series of work and several papers later, what Renee showed was that here we have stress, and we get our activation of the hypothalamic pituitary adrenal axis, and tells our adrenal glands to produce cortisol. And cortisol can bind to the glucocorticoid receptor, and through a non-genomic pathway can induce DNA damage, and that's what she showed. So here we're starting to get into the mechanism a little bit more of how things are happening, and potentially understand how stress hormones can impact immune cells. Also, when I started here at Brighton, a lady called Hazel Hunt, who is the Chief Scientific Officer for Corset Therapeutics, contacted me. And so what Corset Therapeutics do is they create drugs to target the glucocorticoid receptor. This is one of the stress hormone receptors. And mainly they did this for Cushing syndrome. This is patients that have a lot of cortisol. And she wanted to move in cancer, and she'd been working with Susan Conzen and others on looking at cancer. And we know that this receptor may be important with cancer progression and disease, and that higher expression of this receptor, the GR, the glucocorticoid receptor, has been associated with poor prognosis, particularly in patients with solid tumours. And so we've started collaborating with Hazel, and so I just wanted to show you some images here of cancer cells. So here's cancer cells. So what happens is the glucocorticoid receptor sits in the cytoplasm, but when you add cortisol, it moves into the nucleus, and then it can cause gene transcription. But we can block this effect using relacorilant, which is a drug that Corset creates, which blocks the stress hormone receptor. And so we really wanted to look into this a little more. And so Guide was another PhD student in my lab. Actually, she was a master's student, and then she came to do a PhD student in our lab. We were originally going to look, actually, an interesting story, we were going to look at the microbiome in breast cancer. And I wrote a grant. It was a really good grant. And they said, the microbiome and breast cancer, we don't believe it. So the microbiome's everywhere now. But anyway, that was okay. But I said to Guide, do you want to study stress? Because this is what we do in our lab. And she said, absolutely. She wholeheartedly believed in the stress response and what it could do. And actually, what she did was she created these models. So these are breast cancer cells here. And as you can see, you can see them sitting there over time. So we grow these breast cancer cells in a dish. And she just looked at them just for a couple of days. And then she took immune cells and added immune cells in there. And what you can see is these are red immune cells. So the green are cancer, and the red immune cells. It's great. The immune cells are doing what they want to do, and they can start kind of getting rid of that cancer cell. But look what happens when you add cortisol, that pesky stress hormone. Well, the immune cells are trying really hard there to get the cancer cell, but actually they don't really succeed. But if you add relacorilant, this is a drug from Corset, you can see initially they started to get bigger, and we thought, blimey, what's going on here? This isn't good. But actually what it was doing was breaking apart, and then we ended up almost getting rid of the tumour as well. So there was something we could do about this. And the reason we put mathematical oncology there was that we wanted to, I could see this, but we couldn't put a number on it. I couldn't quantify it. And I like to quantify things. I want to see if things go up and down. I like to measure it. I don't like just looking at images. And we worked with some very passionate mathematicians that used to argue all the time, didn't they? But anyway, they helped us put a number on this, and we could identify, and we worked on a model for this, which we published. And then Guide also showed she wanted to look at not only the effects of cortisol on the cancer cells, the immune system, but if you remember, I spoke about cancer cell migration. That's one of the hallmarks of cancer cells. So if we take breast cancer, for example, it's fine if it gets treated early, but if it doesn't, then it moves to those hard to treat places, so the lung, the bone, and the brain. And so we just did a simple assay. This is called a scratch assay here. And so what happens is these are just cells grown flat in a dish, just cancer cells, and you just simply put a scratch in. And what happens, if those cells migrate, the scratch gets less. So it's called a wound healing assay. Now, if you have a wound on your skin, you want your cells to migrate and heal. But if you've got cancer, you don't want those cells to migrate, right? And so so here's our control. If you add cortisol, you see those cells are migrating. So that means that cortisol is telling cancer cells to migrate and move, and that's not what we want. But if we added relacorilant or relacorilant and cortisol, you see it decrease that effect. And she did a ton more work on this, which we are hoping to get published. But ultimately, she showed that these stress hormones can affect not only the immune cell, but how our cancer cells migrate, how they invade. And then she's done a lot of work in the brain that I don't have time to show you today. And Andrew's continued on with that. My final bit of mentoring advice, we're coming to the end, I promise, is that work with fantastic collaborators. And I'm so lucky to be able to say that we've done this. So the question I get asked all the time is, can stress cause cancer? And that's kind of a tough question. And as a researcher that's stressed quite a lot, I hope not. But I'd often say, you know, I don't think it can actually, but I think it can be a contributing factor. And then I thought about it. I thought, well, I need to really look at this. So I worked together with these wonderful ladies. Premal Thaker is at Washington University in St. Louis. Susan Luckendorf is a psychologist in Iowa, and Susan Conzen is a clinician she was in Chicago, she's moved to Texas now. And what we did was we reviewed all the literature, so all the patients reported stress levels and their clinical outcomes, the mechanistic work in cells like we do. And ultimately, it was as we expected. So if there's something else going on, like a virus, a persistent virus, or if there's mutations or sometimes changes in our gene levels, then stress could certainly play a part. But still, you know, there's still a lot to learn on that. And on that front, and talking of fantastic collaborators, this is a lady, Ross Ellis. She's a geneticist at the Institute of Cancer Research. And I gave a talk at the British Breast Group meeting, Ross is great. She said, I've got samples from healthy men and women with BRCA mutations. That means that they have a mutation in a DNA repair protein, which means they can't repair DNA damage if DNA damage occurs. And she says, I've taken serial blood samples and I've taken serial urine samples. Should we measure stress hormones? I'm thinking, great, absolutely, shall we? And so what we did that. Now, it's not a brilliant assay because actually the best way to measure stress in patients is a salivary sample, which is what we try and do now, is a salivary sample or a hair sample. Blood is a snapshot, it's not great. But nonetheless, it's what we had available. And what we found was that the high median levels of cortisol and urinary A2HDG, which is a DNA damage marker, were significantly higher or associated with an increased probability of a cancer diagnosis in these healthy men and women. And what she did was she looked at this snapshot of men and then who developed prostate cancer, not all of them did, and a snapshot of women and who developed breast cancer. And it was those with those higher levels of stress hormones and DNA damage marker that had that increased prognosis. And we've also done some mechanistic work to add to this as well. And so that is ongoing work, and we're really excited about that as well. And on the topic of finding brilliant collaborators, here's the gynae clinical team. I know Sonali's here in the audience up there. And so when a few years ago, here's Sonali Kaushik, Kate Lancaster and Bex Herbertson, and then here's Sarah, PhD student in my lab, and Ketty, a wannabe MD student in our lab actually. And we got together over a bottle of wine and some dinner, and we said, right, we want to do research. And the clinician said, we're really interested in stress, and we want to have clinicians come into the lab. And we were very fortunate to have Sarah Barron, who joined our lab, who's absolutely amazing. And then also we can get samples as well. And we're really proud, because if you remember my interview talk when I came to Brighton, I wanted a translational lab, and we are working on clinical trials now, and also together with Shaw-C, where the first one is the impact of psychological stress on cancer burden and recurrence of ovarian cancer. We're recruiting our last couple of patients now in December, and we'll get the final data at the end of next year. So we're really excited about that. And the second project is just starting with Sarah Becker here, is looking at the impact of stress on endometrial cancer and drug resistance, something very close to my heart, and that we should be recruiting very, very soon. And so what we're doing in this is we're looking at the effects of stress and the glucocorticoid receptor. So here, what happens, what's the patient's journey? So we take a hair sample, and we can look at long-term stress levels if patients are willing to share their hair. We offer them questionnaire. This is with help with Helena and Leslie and Val at Shaw-C. And we look at patients' self-reported outcomes so they can tell us how stressed that they are. We take a saliva sample. So this is good because cortisol's on a diurnal rhythm. So your stress hormones, when you get up in the morning, they're higher levels because they get your fight and flight to go, and then they dip down at night. And so we can measure these. And actually, we did a project with Leslie looking at stress levels. And one lady was disco dancing at night. And we could see her flat line actually go up when she was disco dancing on New Year's Eve. We take a blood sample as well, so I can still look at my all-important immune cells, and then also a tumour tissue. So the bit that the clinician, normally the surgeon, throws away, we can get it and we can grow. I'm saying we can grow organoids. It's not easy to do this, and my lab will know it's nearly killing us, but we're going to do it. Is to grow these organoids outside the dish. We don't have to bother the patient, and then we can treat the organoids with whatever we want, our stress hormones, and have a look and see how they respond. And then just a couple more slides. Now, obviously, with the advent of immunotherapies, this is right on my alley because it's the immune system. There are some brilliant immune therapies. Just a shout out to Marta Valsanelli, who did some beautiful work on how, if we can block the stress hormones, we can improve some immune therapy treatments in cancer. And in cancer, we know immune therapies are playing a really important role. And we always have to show this slide. So this is Leslie and I. We were made up to the nines by a makeup artist, and Caroline Garrett, who's in the audience over there with me. And so Elizabeth Early was very interested, part of the Breast Cancer Research Foundation and a friend of Leslie's as well. And Leslie's covering her eyes with this photo. we don't like this photo. But it is beautiful. And we tested Liz Hurley's stress levels to show how simple it is. So you take a salivary sample, we can measure her stress levels before. She said she was very stressed, and we're looking at her very glamorous. Are you sure, Liz, you're stressed? But yes, apparently she was. And then after I think she exercised or went walking with dogs or something in the park, and we could see her stress levels change. So this was the celeb that came to our lab. So yes, we did have a celeb in the lab, which was absolutely fantastic. But what she did do actually was to promote how important stress was and managing stress levels, which is really important. And something that I really, really want to do is to help patients at the very beginning of their journey to understand how stressful this is, but there are things we can do about it, either behaviourally, through psychology and interventions, pharmacologically as well, through those interventions as well. And a final academic slide. I'm so immensely proud of this. I'm just going to point out these people. This is Simon Mitchell from the medical school, Tim Humphries from the University of Sussex, and Duncan Gilbert from the NHS Trust. And so what happened, Simon was thinking of setting up a cancer centre, and Tim had just arrived from Oxford, and he saw all these people doing cool cancer research, but we're all kind of dispersed around. And so we had this vision to create this cancer centre, and we have the Sussex Cancer Research Centre. This is between all 4 sites, including the NHS. And the idea is we can go from the lab bench and do translational research. If we're together, we can help patients sooner. We can do lots of really good work together. We can share our resources. So some may have fancy microscopes, some may have confocal microscopes, flow cytometers, and everybody is absolutely wonderful. And so we had our launch, and this was at the end of the launch. We had 130 people come to the launch, and this was nurses, clinicians, patients, everybody came, and we were so immensely proud. And this was us at the end of the launch. We were absolutely shattered. I don't think we'd had much sleep. But we were very happy with it. And you can see here, patients are the focus of what we do, and that's why this is a patient here on our symbol. We did steal the seagull from Leslie's group here. But here is the Sussex Cancer Research Centre. We've still got a long way to go. This is just the start. We've just gone through some funding rounds and offered some funding for people, which is really exciting. And we're still building on this, and we've still got a lot of work to do, but it's something I'm, with my colleagues sitting there, amazingly proud of. And I'm so honoured to work with those guys. They're absolutely amazing. And just finally, I did set up my cancer stress laboratory. You're welcome to come and visit any time. You'll see us in the pink lab coats, and you're welcome any time. And finally, just acknowledgements is very important. I'd like to thank everybody in the cancer stress lab, past and present. Brilliant people at the University of Brighton and at the medical school. We are so fortunate enough to be able to have a medical school nearby and work with these fantastic people. Some new collaborations we're setting up as well. And some of our external collaborators that are in the USA and in England. I hope I haven't forgotten people. Wonderful funders, and finally, all the wonderful patients that have volunteered to take part in our feasibility trials. And with that, I will pass on to Andrew. Thank you very much for your time.